Thanks a lot for inviting me to the NBIA Family Conference. And I'm sorry I was not able to attend the meeting in person. Hopefully, I'll be able to see you guys sometime in future. So the focus of my work today, which I'll be talking about, is MPAN, which is caused by mutations in this gene called C19orf12. So basically, MPAN or other forms of neurodegeneration with brain iron accumulation are familial diseases. They're early onset, and there's a wide spectrum of symptoms which varies from patient to patient and disease classes. But the common symptoms include dystonia and Parkinsonism, optic nerve atrophy, and later on in life, the patients can also develop dementia. There is extensive neuronal loss in two parts of the brain called globus pallidus and substantia nigra. These areas are involved in voluntary locomotion. And in addition to the neuronal loss, there is extensive accumulation of iron in these areas. But we should also note that apart from these two areas, which I talked about, there are other areas of the brain also which show neuronal loss, and there is symptoms related to their dysfunction also in these patients. But the most remarkable deficits are related to the structures in the basal ganglia, that is including globus pallidus and substantia nigra. So here is a histopathological examination of a brain that was performed by Penny Hogarth earlier. And this is an image showing some of the findings which we observe in these patients. So this is an image of the globus pallidus, which shows there is marked neuronal loss, and you should observe these eosinophilic pink globules, which are known as spheroids, which are indicator of neurodegeneration. And this blue stain, pearl stain, shows accumulation of iron in these areas. In addition, they have this very interesting finding, which we can see here. This pink structure with a paler rim is what we call Lewy bodies. And there is marked accumulation of Lewy bodies in the brains of these patients. And this is interesting because Lewy bodies are hallmark of sporadic Parkinson's disease. So this indicates that there may be similarities between the disease mechanism for NBIA related to C19orf12 and sporadic Parkinson's disease. So there are around 10 genes that have been identified so far to be involved in NBIA. And one of the challenges that we face in this community is that these genes seem to be involved in diverse biological processes, and we don't have a clear unifying theory for the mechanism of this disease. So as you can expect from the iron accumulation observed in these patients, some of the genes are directly involved in iron homeostasis, including ferritin light chain and ceruloplasmin. Some of the genes are involved in autophagosome lysosomes, and these are organelles which are required for clearance of degraded damaged structures in the cell. Some of the proteins are present in mitochondria, which is a powerhouse of the cell. That is, in addition to doing many other important biological functions, they are required for generating energy for the cell. And other important process in which these molecules are involved is lipid metabolism. So two of the proteins, PLA2G6 and FA2H, are involved in various forms of lipid metabolism. And also pantothenate kinase and CoZ are indirectly involved in lipid metabolism. The focus of our work today is C19orf12, and the function of this protein is largely unknown, and this is what we are trying to figure out. So the model system that I'm using for our studies is Drosophila melanogaster, is the humble fruit fly. And The reason we use it is that this is a very powerful genetic system which has been helpful in understanding a lot of biological processes over the last century. And we have developed a large battery of tools which can allow us to genetically manipulate this organism in a very fast and cheap manner. And over the last years, this organism has been used to understand various human diseases. And the reason is that more than 2/3 of genes that are implicated in human diseases are actually conserved in this animal. So here is just a schematic of a fruit fly brain. It's a simpler brain compared to the human brain. It has around 100,000 neurons as opposed to the 86 billion neurons in human brains. But it performs a lot of sophisticated structure. It has very complicated behaviors. It is and this organism can see, it can smell, it has very complex mating behavior. It can fly, it can perform locomotion, and these are various parts of the brain which are shown here in various colors. So the brain is compartmentalized into different structures, just like our brain, and these structures are intricately communicating with each other to perform various behaviors. So this is a somewhat simpler system, but still it is able to provide us a lot of information about various neuronal processes. So there are two genes for C19orf12 in fruit flies. Both these genes are homologous to C19orf12, and we have taken the CRISPR-based approach to delete both these genes. So the first gene is CG3740, and the second gene is NASO or CG11671. So we have generated a deletion of 134 nucleotides, which removes very important parts of this protein. And we believe that this is a null mutant. And the another one, NASO, has a 657 nucleotide deletion, which also removes the entire gene. So both of these genes we are quite confident are null mutants. So we first examined the longevity lifespan of these flies. And here on the y-axis, you can see the percentage of flies that are surviving. And on the x-axis is the timeline, how many days they're surviving. So the usual flies survive up to 80 days. So basically, you can roughly think that an 80-year-old fly, 80-day-old fly is equal to an 80-year-old man. The medium lifespan of the wild-type flies is something around 55 days, whereas what you can see on the left side is that the median lifespan of the NASO mutant is around 38 days. So it's markedly diminished as compared to wild-type. And the double mutant has similar effect on lifespan, whereas the CG3740 mutant has relatively subtle defects in longevity. Then we performed a climbing assay or locomotor assay in these mutants. And we have a very standardized assay for locomotion in fruit flies. Basically, we take a culture of fruit flies and we tap them down to the bottom of the culture of the vial. And they have this innate tendency to move up, climb up the vial. And we measure how much distance they have traveled over a given amount of time. So I want you to focus on, there are going to be 6 vials. Vial 1 and 2 are going to be one genotype, 3 and 4 another, and 5 and 6 another. And you'll see that there is significant differences in the locomotor ability of these flies. And then I'll talk about what is what. So basically, this is showing you, this is a quantification of that data. So in here, the vial 1 and 2 was the CG3740 fly. The 3 and 4, which was in the middle and had diminished locomotion activity, was the NASO mutant. And then on the last 2 vials were the wild type. So here's a quantification of the data, and it shows that there is significant loss of locomotor activity in the NASO and double mutants, whereas in the CG3740 mutant, the loss is relatively subtle. So basically, the NASO and double mutants have both diminished longevity and diminished locomotor activity. We also examined if they are showing neurodegenerative phenotypes. So basically, this is the eye of the fly, and we're looking at the retina here. And you can see these pink columns here. These are the rhabdomeres, and which are very beautifully organized in the wild type animal, in the normal animal. And you can see all these large vacuoles in the mutants, indicating that there is severe retinal degeneration in these mutants. Also, this is a brain of the fruit fly, and here you have on the left side, you have the wild type brain, and here you have the mutant brain, and you can see that there are all these vacuoles in these brains, indicating that they have neurodegeneration too. So basically, these flies have various phenotypes: locomotor deficit, lifespan deficit, and neurodegeneration. So our main goal with the using Drosophila as a system to understand this protein is to understand the basic cell biological and biochemical function of this protein. And we believe that because we can manipulate this organism so quickly, we can this can be very helpful in providing us information in that regard. And then we can take that information in higher systems, for example, in mouse or human cell culture systems, to understand those functions in a more appropriate way. So we performed an experiment. It's known as the RNA-seq experiment. And basically what we do, it allows you to quantify the expression of the all the RNAs inside the cell and compare it with the wild type. So basically we have the wild type animals and we have the mutant animals, and we are looking at what genes are either expressed at a higher level or lower level in these mutants. And that could provide us information about what the basic biological function of this protein is. So interestingly, a lot of the genes that we identified through this approach were actually involved in lipid metabolism. And that got us interested because many of the genes that we talked about earlier, like PLA2G6, are actually involved in lipid metabolism. So we started examining the role of this protein in this pathway in better detail using these experiments, and I'll talk to you about it. And I'll walk you through. There are some complex slides, and hopefully I'll be able to provide you good information so that you understand it with clarity what is going on here. So one of the organs where NASO is very highly expressed is the gut. And so because there are lipid abnormalities in or abnormal expression of lipid related genes in RNA-seq experiments, we decided to look at lipid related phenotypes in the gut. And what is interesting is that in the wild type, you can see all these green structures. So this is a dye called BODP, which stains lipid droplets. And so lipid droplets are organelles which accumulate fat or a triglyceride for the cell. And as the cell needs triglycerides for energy needs or for various other biosynthetic needs, they are basically catabolized into various lipid products. So in the gut, you can see that there's a lot of lipid droplets in the normal animal. But in the NASO mutants, it is significantly reduced, and the same can be seen in double mutants. CG3740 mutants does not have a significant phenotype in that regard. We also looked at these phenotypes related in the brain, in the retina, actually. And the difficulty with the retina is that at a basal level, under unchallenged circumstances, you cannot see a lot of lipid droplets in the retina. But there are certain scenarios in which there is accumulation of lipid droplets. So there is one gene, Brummer, and we depleted it, and it can lead to accumulation of lipid droplets in retina. And in that scenario, if you deplete the two C19orf12 homologs, you can see that there is significant reduction in lipid droplets in the retina. So basically, these animals have lipid droplet or triglyceride homeostasis phenotypes in various organelles. So now what we are trying to do is we are following it in the gut because we have a robust system and we think that it is very easy to do, and the phenotype is striking, and we think that we can make progress here quickly about the nitty-gritties of its biological function. And then we can take it to other systems to understand it better and provide more relevant information about the human disease. So we have followed it in gut, and this is just to show that the protein is predominantly functioning in enterocytes, which are cells lining the gut lumen in this context. So if you introduce the wild type version of this protein in the enterocytes of these animals, you can largely rescue the loss of lipid droplets in these flies. So you can see that if we express it in the enterocyte, the number of lipid droplets is almost same between wild type and mutant animal. In another experiment, we performed depletion of this protein using another approach known as microRNA, and we specifically targeted the enterocytes, and it can the depletion can actually lead to the same phenotype as the NASO null mutant. So NASO is required in enterocytes, and then we decided to check what are the broader implications of the deficiency that we observe in the guts of these animals. So this is the fat body. So just like human fat, the Drosophila have the fat bodies, and we wanted to see if there are defects in the fat bodies of these animals. And here is a wild type animal, normal animal, and you can see these large lipid droplets in there. And then if you look at the NASO mutant or CG3740 mutants or double mutants, they have significant loss of lipid droplets in the fat bodies. And in the bottom, this is just a quantification of that. And we confirmed this by also doing triglyceride levels in these mutants. So the other question was, are there other impacts related to triglyceride homeostasis in these organisms? So one of the key players in neurodegeneration is oxidative stress. And lipid droplets store triglycerides, and triglycerides are composed of fatty acids. So the theory is that under circumstances where there is increased oxidative stress, triglycerides sequester the fatty acids and prevent oxidative damage to the organism. So if you have defects in lipid droplet biosynthesis, there would be increased sensitivity to oxidative stress. And that's what we're seeing in these flies. So there is an agent called H2O2, hydrogen peroxide, which causes oxidative stress. And if you treat these animals with H2O2, you see that they start dying. And this is a survival curve. On the left side, you have, on the y-axis, you have the percentage of animals surviving. So in blue, you can see that there are wild type animals and they are gradually dying in the presence of H2O2. But if you look at the NASO mutants, which is in gray, or the double mutant, they're dying even faster as compared to the wild type animals. So they're more sensitive to oxidative stress. And here is a quantification of hydrogen peroxide H2O2 in these animals. And you can see that NASO mutants have higher H2O2 as compared to wild type animals. So both they have high oxidative stress and they are more sensitive to exogenous oxidative stress. So having talked about the involvement of this protein in fly in lipid biology, there is one intriguing piece of data, and this is the expression of the human C19orf12 protein. And what is interesting is that if you look at the expression of C19orf12, you can see it is present in various parts of the brain, various other tissues. So it's a very widely expressed protein, but the highest expression is actually in the adipose tissue. And this, you can see that it is also present in breast, where this is which is very rich in fat. It is also present at very high levels in liver, which is involved in lipid metabolism. So this points us that this protein may have very significant roles to play in lipid metabolism. The question that is still open is that whether this role that is involved in neurodegeneration is relevant primarily in neurons, which most likely is the case, but there is a very intriguing possibility, and less likely, I would say, that something is happening in the periphery in these patients, and it is because of the role of this protein in non-neuronal tissues like adipose tissues that they may have degeneration, neurodegeneration. But these are questions that need to be answered in future in different models. So we wanted to understand where this protein is actually localized. And so we express this protein in Drosophila S2 cells, and we have performed the same experiments in living organisms in the guts. And when we express NASO, we observe that it is present in these small dots inside the cell. And if you stain the cell markers for other organelles like lipid droplets, which is in green, you can see that these dots localize to the periphery of the lipid droplets. And they superimpose with another structure, another staining called calnexin, which is a marker for endoplasmic reticulum. So basically, this protein is the localization pattern is indicative of the presence of this protein in these structures known as endoplasmic reticulum lipid droplet contact sites. So these are bridges between these two organelles, which are required for the transfer of biomolecules between these organelles. And we see a similar staining here on the right side in the guts of these animals too. So NASO seems to be an ER lipid droplet contact site protein in Drosophila, and in future we need to examine it more carefully in various other tissues, humans and Drosophila, and we'll learn more about where it is functioning and localizing. So this is just a schematic showing various proteins which are known to be functioning in the ER lipid droplet contact sites. So this ER lipid droplet contact site protein actually serves as the origin of the generation of lipid droplets. This is where lipid droplets are born out of endoplasmic reticulum, and their dysfunction causes remarkable changes in the composition of lipid droplets, and which can have significant consequences for their function. They can be abnormally formed and can be depleted by lipases in the next step. So the next question we wanted to ask was, so why is there lipid droplet depletion in these mutants? Is it because there is diminished synthesis of lipid droplets, or is it because there is faster degradation of lipid droplets, that is faster lipolysis in these mutants? So here's just a slightly complex experiment, but I'll talk about it and hopefully it's clear to you. So this is a normal fly fed with normal food, and you can see that it has a lot of lipid droplets, and here is the mutant which has very little lipid droplets. But if you treat these flies with high fat diet, you can see that mutant regains the lipid droplets. So it's likely that the problem is not in the absorption or incorporation of triglycerides in the lipid droplets, because high fat diet can suppress this phenotype. Interestingly, if you transfer these flies to normal diet again, within a few days, the lipid droplets are largely gone. So these are suggestive that the defect is due to increased lipolysis in these mutants. So they are taking up fat, but they are burning fat very quickly. And so we wanted to identify molecules which are interacting with these with NASO to gain a better understanding of its function. And we looked at various proteins, various genes for their ability to modify the NASO phenotype. And two of the proteins which gave us significant results were Brummer and perilipin 2. I'll explain what they do. They are related proteins functioning in similar pathways. So here is the wild-type fly. It has a lot of lipid droplets. And here is a NASO mutant fly, which has very little lipid droplets. But in the NASO mutant flies, if you can deplete this gene called Brummer, you can actually suppress the phenotype. They gain back the lipid droplets. Similarly, in the NASO mutant flies, if you can overexpress this protein perilipin 2, they gain back the lipid droplets. So basically, this Brummer perilipin 2 axis is able to somehow suppress the NASO phenotypes. So what is happening here? Why is this happening? So this is what is Brummer doing? So basically, Brummer is known to be a lipase. So Brummer is required for the degradation of lipid droplets. And perilipin 2 is an inhibitor of Brummer. So perilipin 2 suppresses the Brummer activity. So our theory is that NASO is a lipid droplet endoplasmic reticulum contact site protein. And when NASO is gone, perilipin 2 levels are diminished, which can be seen in the left panel. And when perilipin 2 is diminished, then Brummer becomes the lipase becomes very active and starts eating up the lipid droplets. And so this is there is remarkable loss of lipid droplets in these mutants. So now in the broader context, there is extensive interest in triglyceride homeostasis in various forms of neurodegeneration, not only NBIA, but other forms of Parkinson's disease and Alzheimer's disease. There is significant data accumulating that lipid biology plays a very important role in the degenerative processes in these diseases. One of the interesting line of thought, which is actively pursued by a lot of different labs, is that in sporadic Parkinson's disease, the accumulation of synuclein that is happening. I showed you Lewy bodies earlier, which are seen both in sporadic Parkinson's disease and NBIA. And the accumulation of synuclein is being proposed due to defects in lipid biology. And it is assumed that synuclein is a lipid binding protein, and disruption of lipid biology in various forms leads to aggregation of synuclein. So we feel that our findings are in sync with information that is being accumulated in other forms of neurodegenerative disease, and hopefully provide the foundation for more work on a human cell culture system, both neuronal and non-neuronal. And we are also trying to get a deeper understanding of the function of this molecule in fly brains. And we are also trying to understand and identify new molecular players that may be interacting with NASO and CG3740 in Drosophila. So overall, we feel that our findings are provocative and would provide new insights into the understanding of the mechanism of this disease. This is the acknowledgement. I am very thankful to the NBIA Disorders Association for providing us the grant, and this has been very helpful in propelling my work. I'm thankful to Dr. Leo Palank. I started this work in his lab, and most of the experiments were performed by Srijit, a talented postdoc in my lab, and Kristen, Sarah, and Madhuka. So thank